Hi class, my name is Alicia Yergi. Today is July 26, 2021. The time is 1:45 a.m. This is for the Bacterial Morphology, week 3, lab 5. This is the exercise and or the experiment and the results for the differential stains. So I have my setup here. Of course, everything was cleaned, wiped down with a bleach solution, everything set up. We did not have to redo our broths because we kept those, so that saved us an extra 48 hours for this. So again, we're going to use our E. coli, the yeast, and the epidermidis samples for this. I do have my slides here labeled, the dime size circle drawn on the slides as well. So for the first thing that we're going to do is we're actually going to get our E. coli broth, and we are going to open this up, and we're going to go ahead and sterilize this, the lip of the broth, or the tube, as we're going to use this one first for our sample on the slide. And again, this is to sterilize it, and I'm just going to place the cap right here. Okay, and then what I'm going to do, I have the inoculation loop sitting in alcohol right now, so I'm just going to, it's been setting for more than 30 seconds, so I'm just going to go ahead and make sure that these are, or this is dry. Okay, and then I'm going to take the inoculation loop and I'm going to put it in our specimen tube of the E. coli. I'm going to try not touching the sides, and I'm going to kind of get in there and stir it up. Okay. And I'm just going to set this tube back here. I will sterilize that again before I cover it, but I need to go ahead and place this on my specimen slide right here. Okay, and then once you're done with the inoculation loop, you are going to get rid of this. I'm just going to put it right here, but we got to soak it in bleach to clean it. I'm going to put this slide down, and I'm going to go ahead and re-sterilize the E. coli tube because I'm done with this one before I cap it and put it back. Okay, so after sterilizing this, I'm going to put that to the side. So I have to wait for the solution to actually completely dry on the E. coli slide. Once it has dried, I am going to use the little tongs to help me assist. I'm going to slide it across the flame on the top, not directly on the flame, but the E. coli slide 5 times to adhere the bacteria to the slide. And then after that, I'll let it cool. We have 4 different stains here that we're going to be using. We have the crystal violet, the PVP iodine, the decolorizer, and the safranin. So again, once E. coli is completely dried, I'm going to put the slide actually in the staining tray. I'm going to add 1 to 2 drops of the crystal violet onto the slide of on the dime-sized area where I put the specimen. Let that sit for 60 seconds. Rinse it in the sink until it runs clear, and then I'm going to go ahead and use the PVP iodine and put 2 to 3 drops on that same E. coli slide. Let it sit for 60 seconds, and I'm going to rinse it off. And then I will use the decolorizer next, 2 to 3 drops. Let it sit for 30 seconds, and then rinse that off. And then we'll use the safranin last, 2 to 3 drops, and let it sit for another 30 seconds. And then we'll set our slide on a paper towel, which I have here, to completely dry before we set it on the stage underneath underneath the lens to see what we have after doing the stains. Once I'm done with the E. coli, I'm going to do or repeat the steps for the yeast and the epidermidis. Otherwise than that, that concludes this video. I'll go ahead and upload results on the data table. And that's it. Thank you.